that much. I'm getting over being sick again. So, I've been like trying my best to study, and I guess we'll see how it goes. Because I don't really want to push off the practice test again. So I think I'll just take it next Tuesday, I think it was, and see how it goes.
Got it. So where are you on reviewing your content diagnostic?
I reviewed all the questions, but I still feel like I do have to go more in depth on things still.
Uh-huh.
Because there were a lot of like questions, I guess, that I wanted to flashcard that like I haven't been able to fully like review yet. But I was able to like, I guess, first glance at everything, and like find more of the weak areas.
Yeah. So yeah, doing that content diagnostic and reviewing it. Um, we want to do before your first practice test. Your team take your first practice test the 25th fifth next.
Um, I Let me double check. Um, the 21st was the original time, but
Oh yeah, okay, yeah, you're right. So um yeah, what were you going to say?
no, I was just like wondering if you still think that would be an okay date because I work tomorrow, Saturday and Sunday, so I won't really have time to study then. Um, so if you think I should like push it more or just see like where I'm at with a practice test.
Do you have the material like Altius or Blueprint or whatever?
No, I was gonna buy it today.
Okay. So let me adjust that. So this would be the third party full length one.
Yes.
Yeah. Okay. Yeah, I think Altius has a better deal with like 10 practice exams for 200 bucks. So I think that would be the best one to get.
Okay.
And yeah, that's what we can do. Um, so all right. What anything else you want to tell me?
Um, I don't think so. My game plan, I guess, for this session was to go through some biochem questions because we did chem physics last time.
Uh-huh.
Um,
Are there
yeah.
any like lingering questions from ChemPhys?
Not necessarily on the stuff that we talked about last week. I did the like the extra Jack Weston practice questions and it went okay. Like the ones I got two of them wrong. One of them was a stupid like equation thing that I didn't see. Like I didn't use the equation that they gave me in the passage. And then the other one I think was like a little confusing, but the explanation made sense.
You
Um,
there questions that you did after the content diagnostic?
yeah, like the passage that we were talking about after we finished our session, like the additional one. So I was going through that one.
Okay. So do you want to continue with that or
Um,
in terms of what you want to go over to? Oh, well, you said you had biochem questions, right?
yeah, I think maybe go through some of those and see how it goes.
Yep. And you're talking about the biochem questions from the content diagnostic or in
Yeah.
okay.
Um, Let me pull some up. Okay. So one of them that I wanted to go over, would it be helpful if I shared my screen again?
Yep.
Okay. Okay. So this one I was a little confused on.
Okay. Walk me through your thought process.
Well, it I thought it would still proceed under physiological conditions because the activation energy is smaller than the reverse reaction.
Okay.
And so I didn't really understand why that was wrong when they were explaining it.
So you said the activation energy is what, higher for the product to turn into the reactant? Is that what you're saying?
Um, yes.
Okay. Um, so let me see. So whether you're going forward, substrate to, or rather reactant to product, you have the um, picture that I drew on the left, and then vice versa if you're going backwards. But regardless of the direction you're going, you still have to overcome the same activation energy, right?
But would it not be easier to overcome the activation energy if you started with the graph on the left than if you started with the graph on the right?
I guess in the sense that you need a higher, like if this is low like this, it's a different distance from the line, but the only ones that we're concerned with are, you know, enthalpy of the reactants here, enthalpy of the products here, and then this hump is your activation energy. So you should have that activation energy not have much to do with this. Oh, and in addition to that, right, the activation energy is a kinetic thing, right?
Mhm.
And um, so regardless of what that activation energy's value is, it's still going to represent um how fast a reaction's going, right?
Yeah.
So I would say for both of those reasons, because like yeah, if you have, again, like, reactants, product, you can measure the heat of the reactant here, heat of the product here, and then the difference between the two is your delta H. But yeah, regardless of which direction you're kind of like, going from, you still have to overcome the same uh activation um energy. But yeah. So that, as well as just the fact that um this is a kinetic thing that deals with rate, uh like the rate of a reaction. Um, but the question says that if we have some, irreversible, let's say A to B, an irreversible reaction, how can we make it so that it can still move forward? And you can do that by just increasing the amount of the reactant, right?
Mhm.
So yeah, so that's how that should.
Okay. And then so then when you're talking about spontaneous versus non-spontaneous reactions, um and like for a spontaneous reaction, the reactants have a higher amount of energy than the products. How does that play into like activation energy if like you still need the same amount of activation energy regardless of if you're going forward or in the reverse?
So uh now that I think about it, you are actually right in that the activation energy is, if this reactant has high energy anyway, it doesn't meet this hump is not as high compared to if it was, oops, something like that, where the activation energy is higher. If you go backwards. So yeah, so you're right on that. But yeah, ultimately this is just a question that's asking, um oh yeah, and then you also asked about spontaneity. Sorry. So the irreversible steps are the ones that have, a negative value or a very, very low value for um their reaction.
So then, can you explain like their explanation when they use the Gibbs free energy equation, like how that would tie in?
Okay, let's see. Gibbs free energy for a given reaction is given by the equation that where Q is the ratio of products to reactants. When the ratio of products to reactants becomes very small, um, i.e., many more reactants, the RT log Q term becomes very negative and decreases the free energy of the reaction. So if the reaction preceding, all right, one second. So okay, um, thus if the reaction preceding triose isomerase produces an excess of reactant, the reaction may still proceed despite being energetically unfavorable under normal conditions. So these are so when you have your delta G negative, you're going to have something be spontaneous, right?
Mhm.
When you have something that's spontaneous, um, it should follow the low or negative change in Gibbs, and that's what you would see at all of the regulatory sites, all of the sites that are irreversible. So, but yeah, ultimately this is, oh, and then yes, this they're using this equation here, which is like this is a standard Gibbs, and then the temperature and the Q value, and that gives you the actual Gibbs free energy. And yeah, if you just kind of go through the choices, like A says it's coupled to ATP hydrolysis. What do you think about that one?
Um, it wouldn't be able to proceed like without a like external energy source, so it wouldn't be like it wouldn't answer the question
Yep,
because it's not like gonna proceed naturally.
yep. Yeah. So ultimately, we want to look at. So A is essentially saying that we can give it some energy from ATP hydrolysis for it to carry out this reaction. Um, B, why do you think B, like considering what we talked about just now, why do you think B would not be a good choice?
Um, well, all reactions proceed very slowly without an enzyme, and um, I guess like talking about the equation,
Okay.
if it was proceeding very slowly, like maybe it wouldn't be spontaneous, or it could be spontaneous, but it would just like take a really long time. So I guess it wouldn't proceed under physiological conditions normally.
So two things, B and D are similar, right? They're essentially telling you that because if you have a low activation energy, um, your reaction will proceed more quickly. And B is saying like, directly that it would happen very slowly. But regardless of whether it's slow or fast, that's not even what they're asking, right? They're just asking, how can this, um, you know, not completely irreversible step? Because if it was a real irreversible step, you would have it be spontaneous, like delta G that's negative. This is not one of those, but the only choice that would fit would be C because DHAP is like a precursor to um a precursor to like blanking for some reason, but um
Um,
like glycer
glyceraldehyde
uh
3 phosphate.
yeah. So you could treat it as like a Le Chatelier's problem that way. If we increase the amount of that reactant, it should push the reaction forward. So
Okay.
it can overcome that positive delta G um, by simply having an excess of a product.
Okay.
Excess of a reactant.
Got it.
Mhm.
Okay. And then next one. I think I just need like to maybe talk through gluconeogenesis because I've been getting a lot of questions wrong on it, and I don't know if it's because of like the like not knowing the reactants in the product or like if there's something else going on that
Yep.
I don't really understand. And this was just kind of like a basic question that was probably just like a content gap, but I have had issues with gluconeogenesis in general.
Okay. So let's see. So in the shared drive, I have in the PDFs folder a file that says metabolism scanned notes.
Let me find it.
Oh, so it wouldn't show up here because It's
Oh, I was looking in like the folder though, because like this one.
oh, yep.
Okay. And you said it's which one?
Yeah, I have a bunch of file called metabolism. So, oh, what? Oh, yeah.
Oh.
Go down one more. Yeah, it'll be this one. So, so okay. I mean, first of all, if you wanted to know what NAD plus and NADH look like, this is what they look like. And um, oh crap, it's a little cut off on the left. But yeah, so that's what these look like. But yeah, you can scroll down some. Pentose phosphate pathway you can look at later, and we can go to gluconeogenesis. Yeah. Okay. So gluconeogenesis is anabolic, which means what?
It breaks down a molecule, or sorry, it adds a molecule together.
So, um, so yeah, it's like, like the food that you eat can be, like if you like protein and stuff like that after like, lifting weights or something like that, you the stuff that you eat will incorporate itself into like your body. So that's anabolic, like like anabolic steroids.
Hmm.
So what's the opposite of anabolic?
Catabolic.
Excellent, yeah. Um so okay, so an catabolism is most of the stuff that we deal with, like glycolysis and Krebs cycle and electron transport chain, but this is for anabolic. So essentially, oh, and then the purpose is to synthesize glucose from non-carbohydrate precursors like lactate, alanine, and glycerol. And you could see here here in the muscle cell, we have glucose being broken down into pyruvate, and then the pyruvate can turn into lactate in that muscle cell, and the lactate can then go into our blood stream, particularly the hepatic portal vein, which is basically like, what is that, by the way?
Um, that's in your kidney
What?
or liver, rather.
Uh-huh. The HEP is, a sign that it's in the liver, but a portal vein is something that is different from other veins, but but yeah, I'll get to the point here. So basically, the lactate gets travels through the bloodstream, goes to the liver cell, and uh, causes the lactate to turn back into pyruvate, and then ultimately into glucose, which can then travel outside of the liver through the blood to the muscle cell. And you can see here this uh, red arrow here says favored, like, in the direction of lactate to glucose. And also that that's when gluconeogenesis is occurring.
Okay.
Yep. And so the yeah, if you scroll down a little bit more, yeah. So the mitochondrial matrix of the liver cell contains pyruvate carboxylase. And so you have essentially these reactions happening. But ultimately, I guess I should just say that ultimately it's essentially the same as gluco, glycolysis, except it's just backwards. So if you go hit the left button here, um, it'll be called metabolism brain dump.
Mm.
Yeah, to the right. Oh, yeah, that's it. Yeah. So, yeah, let's go down. There will be a page that talks about okay, here we go. So we zoom in a bit down there, yeah. Okay, so in metabolism, you want to be aware of, of course, like what's the purpose of something, how does it do it, and then ultimately where is the uh reaction taking place, right? So here we have lactate, which is what I showed you earlier with the Cori cycle, where the lactate can turn back into the pyruvate. Um, uh, so here, the lactate turns into the pyruvate, which can enter into the mitochondrial matrix. And
Hmm.
also, amino acids can also turn into pyruvate and things like that. But yeah, the pyruvate inside of the mitochondrial matrix turns into OAA, oxaloacetate, and ultimately like malate, and it leaves the mitochondria at that point, and then it gets oxidized into oxaloacetate, and that can then result in PEP. And then I kind of skipped some steps, so I wrote stuff. But then after that, if you scroll down some more, so yeah, there's more of this. And but you see how in this step, like this last step here, we have the endoplasmic reticulum being like a compartment. So like the glucose 6 phosphate would enter into the endoplasmic reticulum and turn into glucose.
Okay.
So,
Um, I had a quick question, like at the beginning too, when you're saying like lactate can turn into pyruvate and like amino acids. Is there like anything else that would also turn into pyruvate,
uh,
like that they could ask about?
yep. So before I get to that, so here in the cytosol, we have glucose turning into glucose 6 phosphate, right? In the first step.
Hmm.
So we see over here that the glucose 6 phosphate could also turn into the glucose, right?
Yeah.
So it's a bit of an issue, right?
Um, yes. And like, is this one of the steps that like differentiates between glycolysis and gluconeogenesis from the enzyme?
Yeah, so going down, like glucose to glucose 6 phosphate will be glycolysis, and going up from glucose 6 phosphate to glucose would be um, gluconeogenesis. But um, but the thing is that if this is like in like a single location, this is called a futile cycle, Futile, because every time you make glucose 6 phosphate from glucose, the glucose 6 phosphate turns back into glucose.
Mhm.
So that's not something we can do much about, but that's why the location matters. So the glucose 6 phosphate, or rather the enzyme that turns glucose 6 phosphate, I'm going to just label it as A, enzyme A, that turns the glucose 6 phosphate back into glucose, could be present, enzyme A, in that endoplasmic reticulum, causing the glucose 6 phosphate to turn into simply glucose. So,
Okay.
mhm.
And so like the ER wouldn't have the enzymes for glycolysis.
uh, correct. The liver has, I think, glucokinase, which is a slight difference from the hexokinase. But yeah, so basically this enzyme will just be located in this compartment to avoid a futile cycle.
Okay.
And I think you had another question or no?
Just the one like from before about the lactate. Um, like besides lactate, is there other molecules that like we should know that would turn into pyruvate?
Oh yeah. So yeah, go to the first page on this. So here we go. So proteins can be broken down into amino acids, which can then undergo deamidation to give you your alpha keto acids. And these can turn into either like stuff like intermediates in the TCA cycle, pyruvate, or acetyl-CoA. Okay. Next,
Mm-hmm.
carbs can be broken into glucose, which can turn into pyruvate, which then can turn into acetyl-CoA and, enter into the Krebs cycle. Fats can be broken down into glycerol and fatty acids, which can further break down into acetyl-CoA, and also the glycerol and fatty acids could turn into pyruvate, which would also then increase the amount of acetyl-CoA. So basically,
Mm-hmm.
so all of these biomolecules that you like ingest, let's say, like it can only be these three, right? Protein, carbs, and fats. But they all produce, I wrote here in red, they all converge at acetyl-CoA. That's something that they always turn into. So what that means is if they're making acetyl-CoA based on that, it will affect the breakdown of it for the purposes of making energy. Because the acetyl-CoA goes into the Krebs cycle, all that stuff happens with the electron transport chain, etc. So,
Okay. And then while we're on the topic of metabolism, I was also going to ask about the pentose phosphate pathway and like the PDH. So would we be able to go through those as well?
yeah, you could open up that previous.
Yeah. Um,
Okay.
I tend to get like the pentose phosphate and PDH complex like mixed up, um, especially for the purposes of them, um, and like going between different pathways.
Yep. So I sent you an image in the chat to answer your other question about how do you know, what breaks down and can be incorporated into the um, into our metabolic processes. So you can see that certain like yeah, the glucogenic ones, the ketogenic ones, and the ones that are both glucogenic and ketogenic. So, those non-carbohydrate precursors could be something from these, but yeah, you still want it to be a small molecule. So the ones that I listed, like, yeah, the three that I listed are good examples of that. Okay, so pentose phosphate pathway purpose is to generate reducing power via NADPH and 5 carbon sugars like ribose 5 phosphate. So you know how DNA, right, requires a ribose sugar, right?
Hmm.
And also, like the electron carriers, like, NADP, NAD, NADH, etc. But yeah, okay, the location's in the cytosol. And it's essentially an oxidative way to form 5 carbon sugars. Okay. Now, this, form on the left. Oh, and then by the way, do you know that like the most stable type of glucose or the most stable like um version of glucose, like, in our bodies is the cyclic one, right?
Yeah.
All right. Yep. So that's here all the way on the left, glucose 6 phosphate. Predominant form in solution is the ring, and it undergoes a reaction. What type of reaction?
Um, Reduction.
Exactly. And when it comes to enzymes, you should know that dehydrogenases are oxidoreductases, which means that they um, deal with redox. So, okay, so now we have the glucose 6 phosphate turn into this thing here, 6 phosphoglucono, I believe that's lowercase delta, lactone. What's a lactone?
Um, I get confused between lactones and lactams, but they're like cyclic, and I think one of them has an amide in it, and the other one doesn't. But I get them mixed up.
What does your gut tell you?
That the lactone has oxygen, and the lactam has nitrogen.
Uh-huh. So I don't know if I told you this before, but like you a lot of times you can kind of figure things out about the definition of a word based on the name. A lactam ends in am.
Mhm.
And then what else ends in am? Or rather, what else has am in it?
Like an amide or an amine group.
Exactly. So you get your yeah, so your lactam will just be the ring that incorporates an amide in it. And a lactone is a ring that incorporates just an ester into it,
Okay.
right? So, okay, then we have some hydrolysis taking place to break the ring to give us our 6-phosphogluconate straight, ring structure. Sorry, straight chain structure. And yep, and then as it goes down, another redox takes place, giving you this beta keto intermediate, and then a decarboxylation takes place, which leaves us with ribulose 5 phosphate, which could further isomerize and it can ultimately turn into the ribose 5 phosphate, which is the one that you're used to seeing in things like, nucleotides and stuff. So the ribose 5 phosphate can then be incorporated into RNA, DNA, um, ATP, NAD, and likely also like FAD, stuff like that also, but I just wrote these. So yeah, again, for metabolic pathways, you want to know like what I write essentially at the beginning of these. It's like, okay, this is what it does, and what else did I say? Like, oh yeah, like this is what it does, like the purpose, the location, and yeah, pretty much that. Yeah.
So is this in between um glycolysis and like when does this typically happen?
So this will happen if you like uh deplete your because we store glucose as glycogen and we can store that in our muscles. But once that glycogen has completed to be finished getting broken down, it can then start the gluconeogenesis process to make more glucose to break down for energy, essentially.
So would this happen before gluconeogenesis then?
Which part? Which one before?
Like the pentose phosphate pathway in general, would that happen before gluconeogenesis?
Oh, I mean, so the gluconeogenesis and glycolysis and all of that stuff are dependent on like the circumstances, whereas the pentose phosphate pathway simply keeps making things that are like not to make like things that are not used to make energy. Like for instance, like glycolysis is the breaking down of like a sugar like glucose and like deamidation, is the breakdown of like amino acids. And you have a lot of things that you can break down, but for the pentose phosphate pathway, since it's the only one really like that we're looking at that's anabolic, it's building things up. So
Mhm.
it's not going to really be related to oh, and it doesn't build up like the proteins or the fats or the carbs, but it builds up the sugars that are involved in nucleotides as well as things like, NAD plus and stuff like that.
Okay.
Hmm.
Would you recommend knowing all the structures of the intermediates? Like, do you think that would be something to focus on or like focus on higher yield content more?
So it depends in how you approach it. If you first of all, you should be able to like maybe like draw a molecule or recognize it. So like and one example would be like you should be able to, you know, from memory draw like the straight chain form of glucose, the epimers of glucose, and the ring form of glucose. So for the most part, like I wouldn't want you to memorize just what the structures look like, but ultimately, what are you doing in the structure? So for instance, here in the glucose 6 phosphate, I see that we have um over here. What do you think this functional group is? It's one of the tricky.
Um, uh, is it a
Oh,
acetyl?
perfect, perfect. Yeah, that's one that doesn't usually come to mind for people. And by the way, there's in the topic of like sugars, there's like I think it's like Benedict reagent or Tollens reagent, but basically what they do is they test for reducing sugars and to test if it's positive, the test result, it'll produce like a silver mirror, like or
Hmm.
so a brick red precipitate.
Yeah, I actually had a question on Tollens reagent because I understood like what it was testing for, but not really the process it was going through. But like we could talk about that after, I guess.
Yeah, yeah. So excellent job in recognizing the hemiacetal here. Um, Now it also is a secondary alcohol, right?
Hmm.
And I don't know if I went over like this generalized biochem like redox kind of pathway, but if you have a an alcohol group, it could be oxidized into a ketone. Okay.
Yeah.
So I'll show you like a little later, like what I mean in terms of like the PDF. But you should be so like what so I don't want you to specifically memorize these structures, but you should just be able to see, okay, we have our hemiacetal here and we have our lactone here, but really what changed is just, the carbonyl here. So that's what we're adding, and that should make sense because if you oxidize a secondary alcohol, you'll get a ketone, right?
Hmm.
So we can see that here. And then the next thing that they do is they're adding water, and the water could break apart the ring. And sorry, yeah, break apart the ring to give you your straight chain form. And then there's a bunch of like other stuff that happens that aren't the most important, but this last thing, the ribose 5 phosphate, would probably be good to be able to draw. Um, so yeah.
Okay. And when the water is being added, it's it's attaching to which part of the molecule? Um, like which carbon or oxygen rather is it adding on to?
Oh, so it's breaking apart the the ring. So it's basically doing, I think it's gonna be doing it at, let's see.
Like would it be at can you see my mouse actually like on the screen?
Yep.
Okay. So would it be breaking like here between the like the one oxygen and then the rest of the CH2?
Uh-huh.
Okay. Yeah, I have trouble seeing I guess different structures like of the same molecule. Sometimes it's hard to visualize.
Oh yeah. Yeah. That could totally be um a bit tough, but I would say I guess I'll think about like how to make that more um yeah, more normal about, yeah, that last or the hydrolysis step. But, you know, the very least, you could see, hey, the reactant has this bond, and that's precisely the bond that is being broken here through hydrolysis. So, so my guess is I could even kind of go backwards. I could say, like, okay, this is making a bond here to form that ring. But yeah, ultimately you're hydrolyzing, this bond here.
Okay. And then I guess last metabolism question would be like the PDH complex. I had a question on that from the diagnostic. I don't know if it would be like helpful, but let's see if I can find it. Oh yeah. This was just a question on like reactants and products of um oh wait, that's pentose phosphate. Hold on. 105. Oh yeah. Also, I know like there's multiple enzymes involved in the complex, but I guess I get the names confused and like what they're doing, and maybe if there's a way that like you could explain it, that would be helpful to memorize.
So anytime you see a dehydrogenase, what type of enzyme would that be?
Redox.
Huh? So oxidoreductase, right?
Mhm.
All right. Now, what would a transacetylase do?
It would move an acetyl group
Good,
from one molecule to the other.
good, good. Now, what what can you tell me about the pentose phosphate pathway?
The pentose phosphate pathway.
Oh my God, what am I talking about? All right. So,
Yeah, it's like confusing.
yeah. No, I apologize. I didn't have
No, it's okay.
the best sleep yesterday, so it's like I'm almost like, you know, falling over, but
That's okay.
um, so the yeah, so go on with what you're
Um, well, in pyruvate dehydrogenase complex, there's, I believe, four different enzymes. Um, I think two of them are dehydrogenases.
Mhm.
Um, but this is one where like I kind of get stuck on what it does
Mhm.
and like the purpose of it. I think it takes pyruvate and it like does it break it down into acetyl-CoA? Like so that I can go into the next pathway. Um, yeah, that's about it.
Yep. So you're going to have your what we're doing here, big picture view, is we're taking our pyruvate and turning it into acetyl-CoA. So remember how it showed you in that PDF how all these biomolecules, when you break them down, converge at acetyl-CoA?
Mhm.
And that's because the acetyl-CoA, in our mitochondrial matrix has that, well, undergoes Krebs cycle, but also the electron transport chain, which, brings in a ton of ATP energy, right?
Yeah.
So if you start with pyruvate, you end up with acetyl-CoA. Think about what's different. What could be different here? Um, Meaning like, so I guess like, how many carbons does pyruvate have?
Um, 3,
Okay. What about acetyl-CoA?
2.
Hmm. So one of one of the first steps is pyruvate turning into CO2, or I should say that, yeah, one of the first things in the the pyruvate dehydrogenase complex is that your pyruvate gets turned into CO2. Now, for the longest time, I actually like thought that that meant that there's a decarboxylation happening because you're getting rid of a CO2, but you're really doing so the final step in oxidation is to turn something into CO2. So the CO2 that's produced from pyruvate at the pyruvate dehydrogenase, like not complex, but the actual pyruvate dehydrogenase enzyme, is what causes that CO2 to be released.
So
So,
the CO2 isn't being released from like the molecule itself?
it is, it is. It's just that there, if you had a decarboxylase, it can cause the CO2 to leave. But also in redox, if you have like a carboxylic acid kind of group, the next stage in oxidation is going to turn it into CO2.
Okay.
Mhm. So, uh, so yeah, I'll refer you to like one of the PDFs that I have that kind of goes over this generalized pathway. But yeah, pyruvate dehydrogenase does that. You have um you have like another transacetylase that does stuff with like lipase. You also have another uh dehydrogenase that also has to do with like lipase. But ultimately, what you're getting is pyruvate, um, acetyl-CoA, and NADH. And what you're putting in would be, like the CoA that comes out of the acetyl-CoA and stuff for the
Okay.
pyruvate dehydrogenase complex. So that means is that, oh, and by the way, there's three enzymes in there. So so we know that one of these yeah, so one of these has to be, or three of these have to be actually like part of the pyruvate dehydrogenase complex. So um yeah, so and you could also remember like there's two dehydrogenases and one transacetylase.
Okay.
Yeah.
Mm, okay. So kind of like switching topics. I have issues with like transcription and translation and the differences between the prokaryotic version and the eukaryotic.
Mhm.
Um, I guess this question was okay. This one was just like whether you could tell the difference between the two, but yeah, I guess
What's your definition of a prokaryote?
um it's unicellular or it doesn't have organelles.
Ho- Uh, how could it make proteins then?
Um, it can't.
So, uh, so yeah, you're close, but it's really that they are unable to. So tell me what your reason was again. You said that there's no organelles, right? Now,
Yeah,
there are ribosomes in there, but more accurate thing to say is there's no membrane-bound organelles,
okay.
right? So like the nucleus is membrane-bound, the endoplasmic reticulum, the Golgi, and all that stuff. So okay, so prokaryotes, let's see. So let's talk a little bit about these. So like tell me about A.
Um, I mean, I th like I guess if you didn't use splicing, like how else would you modify the transcript
What is splicing?
when you kind of like cut different pieces of the transcript and like place them together?
Huh? So tell me about that. So tell me a little bit more about that, like in the context of like introns and exons. So
Um,
when DNA gets transcribed into RNA, and before the RNA turns into, your mRNA, what happens in this step, like the RNA to the mRNA? There's three things that happen.
well, the exons stay like on the transcript and the introns leave.
Huh?
Um, and then there's like other post-transcriptional modifications that can happen.
So, uh, so typically when we talk about post or pre-transcriptional modifications, we're talking about like to the protein, but yeah, so now DNA is super, super stable, and it's like in the nucleus, it's like a vault or like a national like whatever library that stores all of that like priceless information, and you're not allowed to take the books out. So the book could be analogous to the DNA, but you could take notes on the book, like in your own notepad, and that'll be like the RNA, because it's literally transcription, which means, you're writing something. Um, but then between RNA and mRNA, we have three things that happen. So first of all, what I just said about DNA is true, but that means the DNA cannot leave the nucleus. It's kind of like that blueprint. But on a need basis, your certain genes will be transcribed into RNA to make those, proteins. So that can be adjusted. So yeah, but when you go from RNA to mRNA, you're adding, oh, and then yes, RNA molecules are inherently a lot less stable. And there's
Mhm.
also a lot of enzymes that destroy it. But yeah, a five prime cap to protect that five prime end of the mRNA, and a poly A tail to protect the end of the mRNA. So those are two things. And then the third thing is splicing. And so like what that means is you could have like intron, exon, or I'll do it like this, exon one, exon two, intron, exon three. So during splicing, the introns leave and the exons are spliced together, like yeah, are spliced into the new sequence. And you can also have alternative splicing, which means you could even have like E1, E3, like skipping E2. Like that's, something that could happen as well. So, so yeah, for prokaryotes, A says, yeah, that they rarely use splicing, and that's true because you don't really have many introns for prokaryotes. And then B, like what can you tell me about an operon?
Um, Operons are in the RNA that can help like activate or repress the RNA.
So, um, so I believe they'll be in the DNA,
So,
but when it comes to transcription, what's the difference between prokaryotes and eukaryotes?
um, prokaryotes can transcribe at the same, like can transcribe multiple parts at the same time, whereas eukaryotes can only go one at a time.
All right, anything else?
Um, Prokaryotic transcription is circular
So, a hint.
because the DNA is circular.
So a hint is it has something to do with prefix and then something that starts with a C.
Mm.
Um, the thing that starts with a C is cis, CIS. So what I'm looking for is cistronic, like polycistronic and monocistronic.
Okay, I've heard of the words before, but
Yep.
I haven't reviewed them.
So yeah, so for us, we or eukaryotes, we have like one for each protein, right? We have one uh gene um that gets sorry. So for each molecule of mRNA that makes a protein, it'll be the same and it'll be like so basically what I'm trying to say is that for each when eukaryotes have transcription happening, there could be one gene for each mRNA molecule that you make, right? Whereas in an operon, you can have, like let's say this is the operon, and this is like gene one, gene two, gene three. The prokaryote can transcribe and then after translate more than one gene or protein in that same mRNA molecule. Whereas for humans and eukaryotes, it has to be spread out. And meaning each RNA would be for one gene product. We can't have, an RNA that can turn into or create more than one product.
So then eukaryotes don't have operons at all.
Mhm.
Okay.
Here, I'm putting this image in the chat. If you could share that screen. Are you able to?
Yeah, I can see it.
Oh yeah. Are you able to share it, the screen? The screen? Are you able to share that? Do you,
Hmm?
do you hear me?
Yeah, I can hear you now. I think
Oh,
you
yeah.
cut out for a second.
Got it. Yeah, no, yeah. I was asking if you could open what I sent you like on the screen that you're sharing.
Yeah, I just did. Are you able to see it?
Um,
It's like in the middle of the screen.
I think you're sharing just your Chrome window, and
Oh,
you need to share the screen itself.
can you see it now?
Yes.
Okay.
So, if we look at the bottom first, prokaryotic mRNA, we have in a single like strip of mRNA genes for multiple proteins. You have protein 1, protein 2, protein 3. So that's polycistronic, right?
Hmm.
Whereas the eukaryotic mRNA above has one protein for each mRNA.
Okay.
So that's a major difference between the two. Oh, and then an operon is the bottom, meaning, you know, something that's in a polycistronic um, piece of mRNA.
And that like determines which proteins are going to get translated.
Mhm.
Okay. So then for something like the lac operon,
Yep.
can you explain that?
Yeah. So that's the same kind of idea in which there's a bunch of well, it was like lac stuff, and like I think there was also like I thought maybe there's like a glucosidase or something. I forgot exactly, but yeah, the thing with that is you can see how depending on the environment of, you know, that cell or during like, transcription and stuff, like if there's like I think it was something like if there's or if lactose is present, then it can make, it can turn on the operon. If it's not present, it can turn off the operon. Um, and also in the operon you have like like a promoter sequence, a repressor sequence that can all play a role in, the types of products that you see. So that's yes, the lac operon is just an example of the example of the operon.
Okay.
Mhm.
So then the main differences between prokaryotes and eukaryotes is that during transcription, the prokaryotes are polycistronic while the eukaryotes are monocistronic, and then prokaryotes don't have membrane-bound organelles.
Mhm.
Um, is there like any other major differences, or it's mostly those?
Uh, yeah, you can include the ones that are in this question, right? Because it says all the above. So
Yeah.
prokaryotes, you could also remember rarely uses splicing. Uh, they use operons, and then your answer is true. Also, they carry out transcription and translation um in the cytosol. They do it, kind of like simultaneously. So, yep.
Okay. All right. Um, I guess I had another question on metabolism, but fatty acid synthesis, I was kind of confused on. Uh, for question. Wait, that's not correct. Okay. I mean, I got it right, but I'm still confused on fatty acid synthesis and like what happens and when it's used.
Okay. So all of these choices talk about acetyl-CoA carboxylase. What would a carboxylase do?
Um, would it add or would it add or take away carbon dioxide or a carboxy group? Carboxyl group?
Would it take it away or would it add onto it?
It would add
Good. Um, if it, what would it be called if it removed?
decarboxylase.
And that would cause what to emerge in the products?
Um, CO2.
Exactly. So, um, yeah, interestingly enough, like all of the food that you eat, um, most of, well, the carbons and stuff that you have, like, or that will have left in you, you lose by exhaling it as CO2. So that's a way that people can measure like your metabolic rate.
Mhm.
But okay, so let's see. So okay, yeah, all of these have acetyl-CoA carboxylases. Acetyl-CoA I know is two carbons, and no well, we're gonna need more, of course, I guess. So okay, not the rate limiting enzyme stimulated by epinephrine, inhibited by insulin, inhibited by glucagon. So I guess between C and D, which are kind of saying the opposite thing, that could kind of help you narrow it down to one of those things.
Mhm.
Yeah. And fatty acid synthesis is like pretty like super like this is probably the first question I've seen on this on like, uh very, very long time. So yeah, we really don't talk too much about fatty acid synthesis, but ultimately, I guess let's think about what it means or what it can do. Um, acetyl-CoA. You said that you just guessed on this one?
Well, I mean, I narrowed it down to C and D, but it was I think it was more so based off of like knowing what insulin versus glucagon does or like when it's used. But fatty acid synthesis, I like didn't really know much about.
Huh? So so insulin takes blood glucose and stores it as glycogen and glucagon breaks down the glycogen to turn it into the glucose so you can actually use it. Now, I guess if they're talking about fatty acid synthesis, they're talking about the building of fatty acid. So it means that, and those are really long molecules. So it makes sense that these are all carboxylases because also if you remember, if you do beta oxidation of a fatty acid, something like let's say this is a fatty acid, what you're able to do is each turn of the beta oxidation removes two, carbons at a time, and then you're left with, oops, and then you're left with just this acetyl group, and oh, and then these all turn into the acetyl-CoA. So, yeah, so that's beta oxidation. So now if we're thinking about the opposite of it, fatty acid synthesis, we can see that it can be um, inhibited by glucagon because glucagon is something that's going to store. Oh, and then I should also say that lipids are, a glycerol backbone with fatty acids attached. That's actually an important distinction. So if you're breaking down fats, um, you're breaking them down into like glycerol and fatty acids. So if you're trying to make fatty acids, right, you're doing like the reverse of the beta oxidation. So it would make sense to inhibit that acetyl-CoA carboxylase by glucose because, sorry, by glucagon because you want to start storing things or you want to start creating those fatty acids if that makes sense.
Okay. You said fatty acids were glycerol and what was the other?
Oh yeah, yeah. If you break down, here I can find like a picture. Sorry, I'm just looking for like a good picture here. Yeah, you could go to the next one while I search for this. For some reason, I keep seeing
Yeah.
like blurred images and stuff. Okay. Yeah, I'll come back to that one prior. So,
Okay.
okay. Electrophoresis. So,
Yeah, I have issues with electrophoresis. I guess,
and electrochemistry, I'm guessing.
yeah,
So,
and the different cell type.
so first I'll state that electrochemical cell equals galvanic equals um voltaic equals concentration. And the opposite is your electrolytic cell. Now, the way that I usually uh, explain this is using just a regular battery because that's a lot more, people don't usually look when they think of a battery, think of like two beakers, you know. So let's say that this is a battery, positive end here, negative end here. And if we had a current carrying wire connecting between the two, we should have electrons leaving here
And
to,
that's still chemical energy rather than electrical energy.
oh,
Because
so
I got a question wrong that was asking that.
yeah, um, so in your electrochemical cell, you are converting your chemical energy, which is this charge separate, like maybe I should put like a barrier between the positive end and the negative end. So due to this concentration gradient, um, it that's the chemical energy part, and converting it into the electrical energy part is the electrons traveling from one end to the next.
Okay.
Right now, this happens until what? What do you think?
Until all the electrons are moved over to the positive side.
Huh? So what do you think these signs will be?
Um, Neutral.
Zero, right?
Yeah.
And because it's at equilibrium here, but this is what a dead battery is, right? Because you're taking advantage, it's like you're pulling the positively charged particles and negatively charged particles, which would rather like to attract one another. We're pulling it away, right? And making a barrier between the two, and then making an alternative path to get from one end to the next end um, that the electron's going to go through. So that charge separation is what drives this electrical energy. And charge separation is the chemical energy part. So okay, when
Okay.
what was
No, I didn't say anything.
it? Okay, so now your what do you do when your battery dies?
Well, depending on the type of battery, you either
Let's say like,
recharge it.
yeah, like a lithium ion battery. Yeah, you recharge it. Okay, now that means that we can just, plug this into an outlet. Now, since we are now starting off with electrical energy, right? We're going to convert it into the chemical energy, aka recreating that charge separation.
Okay.
And since we're forcing this reaction to proceed backwards, momentarily the bottom part would have a positive charge and the top would have a negative charge, so that these incoming electrons don't go to the top part. They only go to the bottom part. Okay.
Okay.
And then once enough of that has happened to recreate this original charge separation, it will recharge that battery.
Okay. So when you plug it into an outlet, it be like the charges switch places
Yeah.
first. Um, but how would they like this is kind of a weird question, but like how would they know to that the top is the negative and the bottom is the positive if they were both neutral when you initially plugged it in?
Yeah. So when you initially plug it in, you are forcing that when you plug it in, right? Electrons from the outlet, go through the wire or the cable. So you're introducing these electrons, right? Now, if it since you're introducing these electrons, they need someplace to go. So that's why, it's not going to stay at equilibrium anymore because you are adding these electrons. And if you're running the galvanic cell backwards, instead of doing chemical energy to electrical energy, you're using electrical energy to form that to turn into chemical energy by forcing it to go backwards. So essentially, you're running it backwards, and that's why you'll have the charges flipped.
Okay.
Mhm.
So even though you're adding electrons to like a dead like neutral battery, it would like the electrons wouldn't equally disperse to both sides.
Yeah, in fact, you don't want them to equally disperse. You want to create that charge separation. And yes, you can do that uh, by plugging it in essentially to force that reaction to happen.
Okay.
Mhm.
And that would be an example of a electrochemical
Oh, yeah.
cell.
So this is the electrochemical as well as these terms. Um, this is specifically a concentration cell because you have a different concentration of the um, lithium ion over here. Um,
Mhm.
and then this is your electrolytic cell.
Okay.
Yeah. So overall, and yeah, again, I think I have like two PDFs where I go over this in the PDFs folder, so I'd say to look over that as well. Um, because yeah, it talks more about like redox and how, at the anode you have oxidation and etc.
So then when you're talking about electrophoresis, can
Ah,
we kind of go through that as well?
yep. So things can either be batteries, which store like the equivalent of electrical energy, Or things can just not be anything like that. Uh, but they still need an electric source, so they might use a battery, right? So your gel, when you do gel electrophoresis, that gel is not itself a battery. Like there's no like, none of this stuff that I drew out is happening on that gel. But you do need to plug it into an external electrical source or, um, a battery, something like that. So that means that you're going to end up having your charges flipped. aka because you're now using it as an electrolytic cell. So,
Okay.
mhm. And yeah,
So so normally the cathode is the site of reduction and then the anode is the site of oxidation. And if something's being oxidized, then it would be n- negatively charged that side.
so yeah, so we have down here our anode. Here we have our cathode. And you could see at the anode, there's loss of electrons, and at the cathode, addition of electrons, right? Now, in order for there to be redox, there must be a redox pair, meaning something that reduces something else means that that something else would oxidize that first thing.
Mhm.
So what can be confusing is for the electrolytic cell here, if electrons are moving to because this is still the anode. So how could that be? Because wouldn't the anode be the site of oxidation, aka loss of electrons? The answer is we don't have redox happening here because there's no redox pair, right? Here we have a connection between both ends, like electricity flowing from one end to the other end. But here we just have the electrons flowing to this bottom end. So yeah, so we're not having a change in, oxidation or, reduction, as well as what the charges could be related to.
Okay.
So for this question, when you see electrophoresis, you can ask yourself, like, have you ever done anything like with electrophoresis? Like, did you work in a lab?
Yeah. Yeah, I worked in a lab and then like during class we would have labs on like gel electrophoresis.
Huh?
Um,
Got it. So yeah, when you look at it, it's like, okay, is this a battery? No. Do I need to plug so for those things, did you need to like plug it in?
yeah,
Ran a gel?
to
Mm-hmm.
to like create the difference in charges.
Wait, so you
Well, actually, that was something I had a question on because with electrophoresis, you're only looking at negatively charged molecules. Is that correct? And like not positively charged?
so regardless of whether they're charged or not charged, um, I mean, they would have to be charged because if this is the thing, um, and so first of all, electrophoresis, it's in the word electro, meaning an electric field is applied, and that's the reason why things move, right? So like if this electric field is going in this direction, that means like, by implication that top part is positive and the bottom part is negative, right? So you're gonna have like uh, you know, like your starting bands here, and they can, reach some points up there. So, but yeah, all in all, over here we have an external source of electricity being like for forcing the things that we're testing for in the gel to move based on, I mean, of course, based on their molecular weight, but the first step must be, I think they phosphorylate first or they do something like that just to make it so that it has a charge. Because if it doesn't have a charge, it's not going to move in this electric field. That's
So then I guess like that was kind of my question of like when you phosphorylate the molecules, like they all have to be negatively charged. Like you're never going to be working with a positively charged molecule.
Yeah, correct. So, yeah, so that's going to be the case. I mean, there's technically speaking, isoelectric focusing, which could be like, showing you something in the opposite direction. But yeah, for the most part, just think about, okay, does this require an external source of electricity? If it does, because yeah, if it does, then it's electrolytic. And if it's electrolytic, the charges flip. So electrochemical over here, we see that the anode is negative and the cathode is positive. All right.
Hmm.
And this one is talking about uh, an electrolytic cell where it's reversed. So your anode will be uh, positively charged, and your cathode would be um, negatively charged, right?
Hmm.
This example.
Okay. I'm going to take a screenshot if that's okay.
Yep, and you can even see here, being powered by a battery. This is an electrolytic thing. Because
Okay.
a battery itself, I'll even add that. So these are all the different words that can be used for this.
Well, wouldn't the battery be the electrolytic
The so
or
the battery,
like the battery itself is electrochemical, but if you're using a battery, then it's electrolytic.
yeah.
Okay. Mhm. Okay. I think I had like one. Oh, um, can we go through like muscles and like the action potential of the muscles? Um, I think I get like the order of things mixed up a lot. And with this question specifically, I was confused because I thought in a sarcomere, the muscle, like this, they overlap, which makes the muscle shorten. Like it's not actually shortening itself. Like I thought it's just the space in between each unit that is getting smaller. So I didn't understand this answer.
All right, let's see what's true of sarcomeres. So this might be another like, wording type of question. So let's see.
It was saying that the sarcomeres sit side by side,
Yep.
but I thought they do overlap if the muscle is like moving.
So I drew kind of like this thing on the left with like the ends being the ends of the sarcomer,
Hmm.
and they can contract, but when they contract or when they um yeah, sorry, yeah. So when they contract, let's see if I can draw it like this. So okay, so basically the sarcomeres themselves cannot overlap because this entire thing is the sarcomer, right? Like you could have the like this part overlap with, this part, like when the actin and myosin connect, you could have that
Okay.
type of overlap, but the overlap will be for, that myosin actin kind of overlap, but
Okay.
it
So
wouldn't.
when it's saying that they sit side by side, that's just literally having the two sarcomeres like right next to each other, which they shorten by having parts of them overlap, but like in general, the sarcomere is still shortening.
Yeah.
Like, is that what they were trying to say in the question?
Uh-huh.
Okay. I think I got confused with like which part of the muscle they were referring to then.
Yeah, so here's another picture. So we could see um that up here, the top one is when it's relaxed. And, when you have that cross bridge, you're forming bonds between the thin filament, which is composed of actin, and the thick filament in red, which is composed of myosin, right?
Hmm.
And when a contraction happens, like if I even like zoom in like here, it's gonna look like, oh, whoops. One second. Yeah, so here it's like, let's say, this is the actin, this is the myosin, and like that, it's like this cross bridge cycle, like a myosin head can attach itself to the actin filament like this. So like let's say that, how can I do this? Well, actually, I don't want to do it that way, but you're gonna get, so okay, on like okay, my this arm is the myosin, and this arm is the actin. And so what can happen is the myosin head, which will be my finger, can attach itself to um the actin at like a 90-degree angle, but then it does a power stroke which goes from 90 degrees to 45 degrees like this. So you see like my finger is straight up,
Yeah.
but if I bend it like this, you see how this is moving to the left, this is moving to the right, and therefore they overlap. So you're gonna have overlap in your thick filament, aka myosin, and thin filament, aka actin. That's where you'll see the
Okay. And then when ATP binds, it like the myosin releases the actin, and the ATP is binding to the actin, or okay.
sorry. It'll be something on the myosin at the myosin head,
Oh, okay.
but you know what rigor mortis is, right?
Yeah.
What is it?
It's when the muscle is like in a permanent state of contraction and it won't release until like all of the ATP is depleted.
Uh, uh, uh-huh. So that's what happens when someone dies, right?
Hmm.
So, okay, so now that we talked about that, let's see. So this is one sarcomer,
Okay.
right? And then this is the sharp. So the top one is a sarcomer when it's relaxed. Bottom one is a sarcomer when it contracts, right?
Yeah.
So the sarcomer can get shorter during contraction, right? However, the sarcomers themselves don't overlap,
Gotcha.
right? Because I'll use green here. So like this is the like other sarcomer. This is the other one. Same thing here. Same thing here, right?
Hmm.
So the overall length of the sarcomer shortens during muscle contraction, and sarcomers contain both thick and thin filament, so A and B are out. And then C says that they overlap, but they don't overlap. It's just that the myosin and actin overlap.
Okay.
Yeah,
Gotcha.
yeah. And you see how like so many of these types of problems are like nuances based on like the word,
Yeah.
because the key like thing here is overlap versus shorten.
Okay. Yeah, I tend to get tripped up on stuff like that.
Unlike when it's like a nuance of a word.
Yeah, because I think I start to overanalyze things too much and get confused.
Mhm.
Um, I might have. I think the last one I was just a little confused on is just the overall topic of like immune cells and like I guess memorizing each like type of immune cell. I need to see what the question was, but yeah, like MHC 1 versus 2 and like the different T cells. Um, I'm a little like
Mhm.
all over the place with.
Okay. Okay. So the there are there are helper T cells and cytotoxic T cells. Okay. What does cytotoxic mean?
Um, it's like toxic to the cell, like
Excellent.
causes death.
Okay. Now I'll use a subscript C for that. And we also call it CD8. Okay. Now, helper T cells we call CD4. And like something that helps me is the Roman numeral 1. If I multiply that by 8, I would get CD8. Whereas the helper T cell, which is CD4, must use MHC2 because the 2 in that turns into or multiplies the 4 to be an 8.
Okay.
So that's just like a little kind of trick for like the um being able to associate which MHC proteins deal with which type of, type of T cell.
So then, what's the difference between a memory T cell and a memory B cell?
What's the difference between a T cell and a B cell? And I'll be right back. I'm just using the bathroom.
Hmm.
All right. Where where are we?
The difference between a B and a T cell.
Between see that?
A B cell and a T cell.
Oh, okay. So, well, oh yeah. Oh, I asked you that. What do you think?
A B cell comes from bone and T cell is from the thyroid, is
Yep.
it?
So very close. They're all going to still be made in the bone marrow, but where they mature is how they get their name. So
Oh,
they mature, I believe, in the thymus. They would get your, uh, T cell maturation.
okay.
So, okay, now the B cell is when you have your um what is it called? Antibodies, right? The the B cell is associated with making antibodies that like the memory, B cells making antibodies that destroy that pathogen, yeah, through the use of antibodies, whereas um like a memory T cell, like T cells can only kill things using the cytotoxic T cells.
So then what is a memory T cell even a real term?
Um, yep, here. I'm putting this in the chat. Oh, let me clear up my thing.
So what so T cells are not, I don't know why I like thought T cells were part of innate immune response, but so like a memory B cell versus like, well, a memory B cell only does like one thing, but you could have a memory helper T cell or a memory cytotoxic T cell.
Uh-huh.
And what's the difference between like memory and effector? Like, is effector like if they just saw like this pathogen for the first time?
So you said if they found the, sorry, tell me that last part.
Like if they found a pathogen and that was like the first time they're seeing it, it would be an effector helper or cytotoxic T cell. But then if it's the second time they're seeing it, it would now be a memory helper or cytotoxic T cell.
Yep. So essentially you could see how there's effectors. Oh yeah, and this as well. So yeah, you could see that over here that so yeah, if we separate it into the helper and cytotoxic, these ones actually kill, and these ones can help assist in the killing by doing things like releasing cytokines. But yeah, that's something that you can. So basically, the only difference between memory B cell and memory like either helper or cytotoxic T cells, I mean, they both still respond to something that you've had before, like some, sickness that you've had before. It's just that the B cell effector B cell creates antibodies, whereas the cytotoxic T cells would just try to kill the infected cell. And then yeah, over here we have another kind of flowchart type of thing. Um, and you could see like a little bit more detail here. So
Mhm.
uh, yeah, all in all, the the T cells are going to be broken, are going to be killing your cells, your infected cells.
So then, um, a memory helper T cell assists in killing, like it, because they don't release cytokines, like only the effector helper T cell releases them.
Mm-hmm.
So then the memory, like, then how is the memory helper T cell different from just a memory B cell,
Yeah. So
besides like where they matured?
yeah. Okay, that's a good question. So basically, let's see. So the CD4, sorry, the helper T cell, the memory helper T cell can can even recruit. So I think they say maybe over here. Well, yeah. So um, yeah. So the helper T cells can activate other immune cells like B cells. Like you'll rarely like see them like not interacting with one another. But yeah, the memory T cells can actually help activate some of the B cells. Like it kind of recruits everything that it can use. That's why it can also make I mean, it'll be the effector one, but that can also make the cytokines. So
Hmm.
yeah, so that's basically going to give you your long-term humoral immunity with that by creating antibodies from the B cells. So um, there's also the antigen presenting cells with the different MH classes, and once that stuff activates, it can start to produce that immune response, right?
Hmm.
So it's a lot quicker because of the MHC stuff that you have, whereas memory B cells don't use MHCs, they recognize antigens directly. Okay. And when they directly recognize an antigen, they differentiate into um, plasma cells, which are somewhere.
Oh, over here.
Yeah.
Here it goes.
So you could see the plasma cells that it that the plasma cells then can secrete their own antibodies, right? And then we can see how that can also affect all of this stuff here. Um,
Hmm.
and release those antibodies, and yeah, and then I guess that's like the major kind of difference that I can think of. Um, oh yeah, I guess another thing would be that T cells, regardless of what type, or rather, yeah, memory T cells, uh, are particularly useful to protect you from intracellular pathogens, meaning pathogens that
Nice.
have, so like viruses, certain types of bacteria, whereas memory B cells are more along the lines of uh, protecting you against extracellular pathogens. Uh, yeah, and I guess those are the ones that I can kind of think of now. Um,
So, what would be an example of like an extracellular pathogen?
oh, I guess like let's see. I think a lot of like I mean, for some reason I'm thinking of bubonic plague, but um, let's see. check out the okay, so extracellular could be located in these regions, and we have certain examples. You could see how there's viruses, bacteria, protozoa, fungi, and worms. And it's useful to so like so tell me a bit about how an antibody works.
Well, there's an antibody and an antigen, and the antibody has two, like a light chain and a heavy chain,
Okay.
and the light chain, I think, is the top part, and that is what attaches to the antigen, and it's specific for like one type of antigen.
Mhm. So um so it binds directly to the antigen, right?
Yeah.
And that's different from if it gets like bound to MHC, right?
So if it gets bound to MHC, that's like a signal for the cell to be destroyed.
So here, let me put another one here. Okay, so MHC class 1 can, present antigens to cytotoxic T cells. MHC2 can present antigens to helper T cells. Um, and yeah, and then so the MHC is going to be an example of, an APC, which is an antigen presenting unit. So it'll phagocytose the bacteria or the pathogen, and it will display on it um, particular like types of antigens from that pathogen. And then, it can then eventually be destroyed by like the cytotoxic T cells. But yeah, I would say that the main takeaways that we have here are that in let's see. Um, oh, and then that's also the difference between cell-mediated immunity and humoral immunity, meaning the memory T cells or T cells in general can work using cell-mediated immunity, but the B cells can have humoral immunity, which means that they can make antibodies, and that the B cells can recognize the antigen directly, whereas the T cells require the antigen to be presented by by an MHC. And then
Okay.
the B cells kill the pathogens using antibodies, whereas the T cells kills like the cells themselves, uh, since they typically involve intracellular pathogens rather than extracellular. So yeah, those are the main differences. And you can kind of see in the picture, like this red thing, so this is like a virus here, and then this is like some, a bunch of bacteria. But here you can see that it's within the cell now.
Mm-hmm.
So, so yeah, I would just make the distinction of T cells need antigen presentation as well as being used to destroy intracellular pathogens, whereas B cells don't need the antigen presenting uh, antigen presentation to happen. They can bind directly to the antigen, and instead of just directly killing the infected cell, it's going to produce antibodies that will handle that. And
Okay.
yeah, intracellular versus extracellular also plays a large role.
Okay.
Mhm.
Okay. Um, then I think that's pretty much everything. Um, yeah. Okay.
So here, I'm just putting one more image in there. And so we have, I guess, on the top, a B cell with receptors that, resemble antibodies, and then like an antigen on this bacteria. It can recognize that. Um,
Mm-hmm.
but it can also have in the next image below a MHC thing that can attach to the helper T cell receptor. And yeah, and then you end up with either um, the memory B cells being like with that the antibodies attached to it because it remembered last time. But you can also have the plasma cells that don't remember that they were attached to the antibodies.
Okay.
Yep. So, all right. So let's see. What's next for you?
Um, Next would be the practice test on Tuesday.
Okay. And oh yeah, we talked about that. So that's going to be, so you haven't gotten like Altius or Blueprint yet, right?
No, not yet.
Okay. So yeah, I think the Altius test is cheaper. So you can get those, and it's up to you how many you want to get. Because if you're taking your test in March, you might not need all 10. But in worst case scenario, like if you push it back or something, then it would be useful to have all 10. And I think the price range isn't too much more expensive. But yeah, so when you do that, also, yes, I forgot to say this. Don't do the Altius cars. For cars, do like a Jack Weston cars passage.
Okay. Like during the polling?
So, um, yes. So you're gonna take your full length, and um, you're going to when CP is over, switch over to another tab, do a Jack Weston cars exam, which you can get to if you click on like, do you know how to get to it?
I know how to get to the like study Q bank.
Do you see what I'm sharing?
Yeah.
So yeah, you're gonna go to practice exams here. And down here, there's a Los Angeles one, there's a Chicago one. Just choose, one of these and do it, obviously with, timed properly,
Okay.
right?
And that has like 10 different like passages
Yep, yep.
did
So you
already.
do that,
Okay.
and then once you're done with that, you go back to the Altius exam where you finish up where you left, you go back to the Altius exam, and this time you do, the BV section and then your psych-soc section, and
Okay.
yeah, you will use a score converter to get your car score. So, okay, that's that. And then the last thing with timing, did I talk to you about timing checkpoints?
You did
Yeah.
briefly,
So
but
basically,
I might need a refresher.
yeah. So basically, what you don't want to do is constantly look at the clock because that's going to really interfere with like your concentration. So there's three designated checkpoints at which you will look at the clock. So the first one is when you get to question 20, you should have an hour left. Okay. And
Okay.
if you have less than an hour left, just gently tell yourself to pick up the pace a bit. Um, your second checkpoint will be once you get up to question 30, you should have 45 minutes left. And the last checkpoint would be when you get up to question 40, and you should have 30 minutes left. So that gives you three designated checkpoints at which you will check the clock rather than checking the clock, like throughout the test. And also, if any question takes longer or feels like it's taking longer than a minute and a half, make an educated guess, flag, and move on. And ideally, you'll end up seeing that last question of the section with
Mm.
time to spare so that there's no surprises, and then you can go back to the ones that you flag.
Okay.
And also in the first 15 minutes, I don't know if you want to do this, but like I would always like brain dump, on my sheet of paper. So uh so I have that information in front of me when I'm taking the test.
Like during the instructional period.
Yep.
Okay.
So all right. So that's pretty much it. And anything else?
I don't think so. I have another meeting, I think, scheduled with you next week, either like a Thursday or Friday, I think. Um,
I see you for Thursday.
okay.
All right. Awesome. So for today, it should just be 188.
Okay, let me know when you get it.
luck.